Nietzsche, ¿estás en Perú?
No, estoy en mi oficina.
Ah, ok, pensé que estabas en Perú.
No, estuve ahí, no sé cuánto, la semana pasada. Yo he ido como 5 o 6 veces ese año.
Qué bien.
Qué bueno.
¿Ese es tu lugar de campo normal, Mitch, en Perú?
No, es más el lugar de mi novia.
Bueno,
tenemos proyectos con World Wildlife Fund en
¿sí?
Amazonas, en Perú. Creo que tenemos otros proyectos en Perú, pero no sé. Ahora con este proyecto, tenemos proyectos en muchas partes. Acabo de terminar un proyecto en Virgin Islands, en El Salvador. Vamos a empezar algo en Honduras.
Qué
Muchos
bien.
proyectos en Colombia. Madagascar. Pero la idea de trabajar más en África me parece muy interesante.
Sí.
Sí, la verdad que sí. Y aquí, claro, en Canarias estamos aquí al lado y hay programas, ahora están empezando a ver si lanzan programas de intercambio de academia, tanto estudiantes como profesores.
Ay, qué bueno.
Sí, que ahora hay un programa Erasmus que es para movilidad europea. Y hace poco nos contactaron a ver si queríamos abrirlo. O sea, la Unión Europea parece que está pensando en abrirlo a lugares africanos, ¿no? Entonces, pues nosotros dijimos que claro, que por nuestra parte sí. Y
Claro.
estaría bien porque hay un montón de cuestiones acústicas bueno, y en cuestión marina hay un montón de impactos acústicos de los que no se sabe nada y no se hace mucho en la mayor parte de África. Si ya en Europa y Estados Unidos está la cosa mal, y en Latinoamérica, en África pues está igual de mal o peor. Entonces,
Wow.
claro, para cuestiones de sísmica, de sonares militares, todas estas cosas así de ruidos potentes, no hay mucho control. Entonces,
Tiene mucho que hacer.
sí, estaría bien. Y supongo que hay también cosas positivas para hacer, ¿no?
Ok, son las 12:01. Should we begin?
Sí.
Okay.
Okay.
Okay, so welcome everyone to our final seminar series of this semester. We have 2 guests today, so each of them will have about 20-25 minutes, and then we'll have at the end of each talk time for questions. You can directly ask the questions, or you can write your questions on the chat, and then I'll translate that for you. And all I will ask is, before we begin, that everybody mutes their computers so that the only sound comes from the speakers. So first we're gonna begin with Mitch Aide. He's a professor at the University of Puerto Rico and Rio Piedras. And Mitch has been working with plant ecology, biogeography, landscape ecology for many, many years. And I think the past decade or so, he has started a number of initiatives in soundscape ecology. And a lot of what Mitch and his team does is to study the impacts of deforestation and habitat loss in climate change and plant communities and fauna communities in tropical areas. And so today he's going to be sharing with us one of their main platform, which is called Arbimon. So thank you, Mitch, for coming today.
Thank you, Laura. Okay. Well, what I'd like to do today is tell you a little bit about the platform that Laura mentioned, and then go into some details of what we call soundscape analyses. The Arbimon platform, it was developed, I think it was now almost 16 years ago. And the idea was to create hardware and software to improve biodiversity monitoring. My background is I'm a tropical plant ecologist. Before we started this project, I did not have any experience in acoustic biology. And really the main goal of what we were trying to do is just figure out better ways that we could monitor biodiversity in many sites, around the world. And we really, our focus, we started working with computer scientists. And so the things that we were really focusing on was trying to create a platform that made it easier for people to store all their recordings and have tools that made it easy for them to analyze. The first thing that I found out is that, recorders work and you usually get swamped with recordings, and people end up having all their recordings on hard drives and, don't often analyze the majority of their data. And so we really thought it was a priority to figure out how to create a way to make it easy for people to store their data, the recordings, and have access to them easily, that they could share them. And so we built this web-based platform called Arbimon, and it now has I think it's over 3.5 million one-minute recordings in the system, hundreds of different projects from around the world. So that part of the system is just, how to manage all of the recordings. The second part is then what can you do with them? And there's two major components that we have developed. One is giving the user tools to create species-specific identification algorithms. There's no magic in the system. There's no button that you can push yet to identify all the species in your recordings. But we do provide tools that if you know the calls of your species, you can create species-specific algorithms. And then once you have a model that works well, then you can apply that to all your recordings. There are many people that have over 100,000 recordings in the system, and they can now, instead of listening to a couple thousand, they can run the model over 100,000 or whatever. The other part, or the other tool that we're providing, which I'm going to talk about today, is this idea of what we call soundscapes. Many people have different definitions of what a soundscape is. Let me see. The way I like to think about it is that we have many recordings from a site. We often put recorders out for a week, recording 1 minute every 10 minutes, 24 hours a day. So it gives us 144 recordings per day, around 1,000 in a week. And now what we're going to do is we're going to take those 1,000 recordings and aggregate them. And what that allows us to do is ask in this site what frequencies are being used, what time of the day are they being used, how much is that time frequency bin occupied, and then trying to decide, what is the origin of the sound. To do this in the system, it's pretty simple. You have your recordings in the system, you give a name to the soundscape, you define a playlist, so it would be all the recordings from a given site or whatever, and then you can select the time of aggregation. Usually what we're doing is using hour of day, but if you had recordings throughout the whole year, you could aggregate by month or by season. The next thing, there are some parameters that one selects. So the bin size that you're actually going to do the analysis on, and some other aspects here. And you end up with something that looks like this. And even if one hasn't even thought very much about acoustic recordings, you show these figures to people, and they quickly get the idea of what's going on. So in the x-axis, we have hours of day. In the y-axis, we have frequency. And in the z-axis, we're just showing what the proportion of recordings had a peak of activity in that period. And so you can see, the density in each one of those time frequency bins. And now looking at this recording, at these two soundscapes, you clearly see that there are big differences between these sites. In both cases, you see a lot of activity below 4 or 5 kilohertz. You see that there's more activity during the nighttime hours than during the day, but there's a much more activity in Madre de Dios in Peru in comparison with El Verde in Puerto Rico. You see that there's two very consistent peaks of activity in Puerto Rico, starting around 4 or 5 in the afternoon and continuing to 6 or 7 in the morning. That in this forest is the one species, it's the coqui. And then showing the two notes and thousands of individuals around this microphone that are screaming coqui for about 10 or 12 hours during the day. But during the daytime hours, you can see the amount of activity drops down a lot, which is very different from what we see in Madre de Dios. We have insects, so we have birds calling, we have insects calling at much higher frequencies, and we're probably capturing some bats. It would be sort of at the lower range of the frequencies used by bats. But one of the things that I really like about the soundscape analysis is that it captures the whole acoustic community. And so you can go into a site that you really don't know all the species, and it's not focusing on any one species, it's just reflecting the sounds in that area. So one of the things that we did was to see, we did these types of analyses in Puerto Rico, in Peru, and then many sites in Costa Rica. And one of the things that we expected, or we were hoping, is that these soundscape analyses would be a good reflection of what the species richness is in the site. And so one of the ways that we were taking these soundscapes and sort of converting them into an index was just summing up the number of time frequency bins that are occupied. In this case, there are 24 hours a day. There was 256 bins, and I think that gives us around 6,000 time frequency pixels, and we could just count up in this acoustic space what proportion of that area was occupied. And in the case of El Verde, you see it was I think around 12%, while in Madre de Dios or Amacaeri, it was around 35%. And so clearly, these two areas, there's much more activity in Peru in comparison to Puerto Rico. We correlated this with regional richness of birds, with amphibians. And the reviewers of the article asked us to be more specific, and they wanted to know, what was the richness of species that were in these recordings, in these soundscapes. And so we took another month or two to go through and identify the acoustic morpho species that we found in these recordings. And we fortunately came out with the same result, that there was a good, a positive relationship between the acoustic space used and the number of acoustic morpho species. One of the nice things, and we often complain about reviewers' comments, but one of the nice things that came out of doing this, it became very clear that the majority of, at least in these tropical forest sites, the best predictor of species of the acoustic space was the diversity or the richness of insects. So, there's certain biases using this type of analysis, and one of the things that is clear is that the insects are playing a very important role in our analysis of the amount of acoustic space that's used. So another way that we've used these types of analysis of soundscape is to see how a community changes across the elevation gradient. And this was done as part of a PhD thesis of Marconi Campos. And he was looking at, he put 60 recorders out on El Yunque, which is the national forest here in Puerto Rico. And this was pre-Hurricane Maria. And then he looked at the soundscapes of these 60 sites. And one of the things that he did, let me go back a slide, is that what he did was use these 6,000, or in his case, I think he used a larger bin size. So he used each of the time frequency bins as a variable and created a matrix of what time frequency bins are being used in his 60 sites across the mountain. And then he was able to use that to create an ordination. So he's taking his 3,000 variables, saying which ones are being used in which sites at which time of the day, how similar are these different sites. And you can see on the left-hand side is his ordination, and he's overlapped the elevation gradient on that. His next step was to do a cluster analysis, and when he did that, there's two main clusters. There's the sites that are less than 700 meters, and those are above 700 meters. The next step, what he did was extracted what are the indicator species, or the indicator, in this case, time frequency pixels. What are the pixels that are distinguishing these two groups? And then what he did, which I thought was interesting, he made a new soundscape, and this soundscape was where's the differences between the lowland and the highland sites. And so that's fine. You have all of these time, okay, there's more of this frequency is getting used at this time of day in the lowlands and the highlands, but that wasn't very satisfying. So what we have in the RBMN platform is a tool that allows you to go in and extract the recordings that are contributing to these peaks of activity. And so he went into these indicator pixels and tried to put names on them, or species. And so here you can see on the left-hand side is the low elevation sites. And one of the things that first came out, if we look at the higher frequency, so there were some bird species that was unique to the lowland sites, and we didn't find it at the higher elevations. If we go down in frequency, you can see there's a couple of species of frogs that are around 700, 750 hertz that were unique to the highlands. If you go down, then there's a lot of insects and frogs in the lowland sites And you get the idea that this was a nice way of being able to go from the whole acoustic community down to individual species that were distinguishing the acoustic community in the lowlands and the highlands of this area. Another use was, which I think was really interesting, and this was another PhD student. His name was Paul Furumo. He was doing his PhD on oil palm in Latin America. And it to me, one of the nice things about acoustic monitoring and doing these soundscapes analyses, it gives people the opportunity to include a biodiversity component in their thesis, even though they're not really experts in this area. Paul was doing his PhD in an environmental science program. His first chapter was a land use study of looking at how oil palm is expanding throughout Latin America. His third chapter was looking at certification and sort of the social economic aspects of certification of sustainable oil palm. But one of the things that he did, was able to add in a biodiversity component in his thesis by using the recorders and looking at soundscapes in and around oil palm landscapes. He did this in Colombia, and there's three main areas of oil palm production in Colombia. One is in the north, one is in the Rio Magdalena, which is, but the northern area was really dominated by bananas for many, many years. And recently the bananas have been replaced by oil palm. The moist forest in the Rio Magdalena, the center area, was pretty much forest and a lot of cattle pastures. And then the third site was in the Llanos of Colombia, so it's on the eastern side of the Andes, and these were savannas and gallery forests, and many of the savannas have been converted into oil palm plantations. The results of what I'm going to show are from the northern area. And what he did was he put recorders out in forest patches, and there's not a whole lot left. There's some riparian forest in oil palm, and then looked at some of the other types of land uses in the area. And that includes pasture, rice, and banana. He did something very similar to what I described before. He used the time frequency pixels as information to create this ordination. And one of the first things that comes out, you can see along the first axis that there's a clear separation of the forest and riparian forest from all the production sites. So we have on the left, forest and riparian, and on the right, we got all of the agriculture. We have pastures and rice and oil palm and banana. The interesting thing that came out of the analysis, we look at on the second axis, the production land use that was most similar to the forest sites was the oil palm. 5 minutes before we started this conference call, he just got back the reviews of this manuscript, and so hopefully it will be published soon. But the idea here is not too surprising. I mean, the important thing that there's a very large difference in the acoustic community between the agricultural sites and the forest sites. But when we think in terms of structure, of these agricultural uses, the rice and pastures are very open areas. Banana is very, how can I say? Obviously it's monoculture like oil palms, but oil palms have a structure of and an understory that is probably a little bit more similar to the forest sites. And that may be why we're seeing that they're a little more similar in terms of their acoustic community to the forest sites and compared to these other agriculture. So that's just a quick overview of how we are producing what we're calling soundscapes, how we're producing them in the 3 examples of how we're applying that to different questions in different parts of the world. And with that, I'll stop and take any questions.
Thank you, Mitch. Does anyone has any questions? You can type your questions in the chat too, so I can read them. You guys have any questions? Mitch, are you, how many, you guys also have indexes of biodiversity, right? Implemented in Arbimon. Are you planning to add more or?
No, really, our experience with many of those diversity indexes, at least in terrestrial systems, we don't see that they're not very well correlated with species richness. That's why we were interested in trying a different approach and using these soundscapes as a way, because they clearly are capturing all of the acoustically active species. And our first paper showed that at least within a forest sites, there is a very good correlation between the acoustic area used and the number of species. The next direction that we're going to take is to try to develop convolutional neural networks that will allow people to do, again, give the user tools that will allow them to do multi-species identification in their recordings. So instead of having to make a model for every single species, what we're trying to do is develop tools that allow people to do identification of multiple species in individual recordings.
Okay, Mitch, there's a question from Nicole. She asks, it looks like there isn't much of a reduction in diversity of found plantations. Is it true? Is this true?
Oh, yes. Yes. Let me go back. No, I mean, I'm sure one of the criticisms of this paper is that the analyses were left at this level. So we did not go into details to try to identify all the species that were in these sites. But one of the things that was clear doing this analysis was using the acoustic space used indices kind of, I don't want to say falls apart, but the area with highest use were pastures and rice, and that's because there were lots and lots of insects and frogs in these systems, and those species use a lot of acoustic space. So to me, one of the things I think people should be careful if they decide to do something like this is within a forest we saw a nice relationship. But given that different groups of species use sound in different ways, in a sense, you can think, there might be a lot of birds in the forest, but their calls are sparse, and it's not this continuous sound of a cicada or a chorus of frogs. And so, in the sense, the index did not help us a lot in this study, but certainly using the soundscape, doing the ordination, I think some very interesting results came out. The next step would be to continue with this and try to put names on those peaks of activities.
And there's one question by John that he says, by just counting time frequency bins, are you concerned that animals with low frequency calls with many harmonics may be overrepresented?
That's certainly something to consider. Yes.
Yes. Okay.
I mean, one of the things with the recorders that we're using, you know, it's not a shotgun recorder. We're now, this study was done using Android cell phones and an external mic. Now we're using audio mods, and there's certainly some species or individuals that get very close to the recorder that, you know, you'll see the harmonics, but in general, I don't think that that's a very big problem. And again, one of the things that we're doing here is that we're aggregating, you know, 1,000 recordings or 10,000 recordings in a single soundscape. So, you know, a couple of recordings that have a lot of harmonics, you would think would probably the effect would get washed out.
Right. Okay, any other questions here? Thank you so much, Mitch, for your time and your presentation.
Thank you.
Yeah, we're now gonna move to Natacha. We're gonna take a couple minutes to get Natacha set up. Thank you, Mitch.
Bye-bye.
Bye.
So I'm here. Do you hear me?
Yes.
Okay.
Let's just
I can share the screen.
Yeah.
Here it is.
Yeah. Yeah, and can you? Yeah, exactly. Perfect. So, for everybody else that just joined us, please make sure that you mute your microphone so that we can only get Natacha sounds. Natacha is a researcher and professor at University La Laguna, that's in Canary Islands, right?
Yeah.
Yeah, Canary Islands, and she is doing really cool research on studying the deep diver marine mammals. She works on understanding the niche of foraging behavior of pilot whales and big whales. So I'm not gonna take too much time from you because I think I want to hear what you're gonna share with us today.
Okay, thank you. Thank you also for inviting me to be here, and I hope that everybody will start being surprised about my pronunciation very soon, and I'm able of explaining well the message of the presentation. The Canary Islands is an oceanic archipelago that is very close to Africa. It's only 100 miles from Africa and something like 1,000 miles from Spain, from mainland Spain. And we are lucky that there are resident populations, year-round populations of at least 5 species of deep diving whales, including short-fin pilot whale, 3 species of big whales, sperm whale, rhesus dolphins. So for these whales, for toothed cetaceans in general, the use of echolocation is essential to mediate foraging in the dark niches that they are using in the dark habitat of the deep waters. And they also use acoustic signals, like other cetaceans, for communication. So by doing sound analysis, we can extract time budgets of their social and foraging activities. Foraging is quantified in 2 ways, because we can attach tags, as you will see now, to the whales, so we can get a temporal and a spatial location of 2 activities: when they are looking for the prey, trying to find it and choose it, and then when they are trying to attempt it, the hunting event. So all this comes to the behavioral ecology of the whales and how feeding activities and then social and vocal behavior interrelate to make the broad niche, what is called the broad niche of a species. A niche can be talked about like only the foraging niche, or you can amplify the term and use it to refer to how a species is living. And that, in turn, has strong implications on how a species is vulnerable more or less to human disturbance. So here we are going to make a fun comparative analysis of the acoustic ecology of two species mainly, Blainville's victual and the short-fin pilot whale, because both live in the Canary Islands and both have been tagged there. And we will use extra data from Cuvier's victuals from Azores and the Mediterranean. These are our main characters. The short-fin pilot whale is a strong animal that is a bit smaller than the long-fin pilot whale. So the morphotype that lives in the Canary Islands can reach something like 5.5 meters and something like 2,000 kilos, maybe a little bit more. And the Blainville's victual is this, we named this whale E.T. because this here, I don't know if you see my arrow, I hope so. These are not the eyes, these are the teeth that only erupt in the males and get covered by barnacles. They're actually down here. So this is Blainville's victual that is in the same family than the Cuvier's victual. And the Cuvier's victual is a bit bigger. It can be up to 7 meters and 3,000 kilos, while the Blainville's is just a little bit smaller than pilot whales and the short-fin pilot whales. So here we have 3 very deep diving animals that you can only see very close to the coast in the Canary Islands and other oceanic islands like Bahamas or Hawaii. So we use acoustic tags that were developed by Johnson and Tayak in the Busol Oceanographic Institute back in 2003. And they are like a telephone has been reduced with the time, and they attach with suction cups to the animals. They have a hydrophone that you can see here. Sometimes they have 2, so they can record in stereo, and they have electronics that can record tons of things. So we will go a little bit more to what they can record in a while. First, to attach them, we do it with respect. We don't approach the animals more than 3 times an individual on one day. We approach slowly with a long pole, and this is Mark, the father of the tags, and they get, as we said, attached with suction cups. So they make an intensive short-term recording of what the whales are doing. And it's very intense because you get the depth profile. This here on top is the depth of one Blainville's victual, and then you have the heading, that is rumbo, the roll, and the pitch. And from the pitch, you can get the fluking rate. The pitch is the inclination of a whale. So the whale is going up and is fluking, and that makes small indentations in the fluke, in the pitch rate, and you can get how often it's fluking. So there are some raw data that one has to analyze to get derived data that are giving you information at a very high resolution of what the whales are doing. And then the sound data, that is why we are here. So the acoustic data, we analyze it in context. So here, for example, we have the depth of the whale. This is at nearly 1,000 meters. And we have here clicks and buzzes, but not only of the whale that carries the tag, also of whales that are in the social group, in the vicinity. So the first problem is to separate the vocalizations of the whale that carries the tag and the whales that are in the surroundings, in the social group. Of course, here we can also detect other sounds that are in the environment, such as boats, and we have made the studies of the impact of shipping on the behavior of whales, sonar, and there have been studies, not by us, but by other people who also use tags, and they have studied how the behavior of the whales changes when they are exposed to sonar, and many other things. So we detect the clicks of untagged whales, non-tagged whales, because they are mainly ultrasonic. So it will be these clicks here. This is an spectrogram, as you probably have seen many in these tags. This is time, this is frequency. So the clicks that go down in frequency are the clicks of the whale that carries the tag, because the low frequencies get transmitted through the body of the whale. And also because the clicks of the whale are directional. They go towards the front. So in the front, you have the ultrasonics, but there are artifacts, there are machinery noise of the producing of the clicks that get transmitted also backwards and in a more omnidirectional way. So those get recorded by the tag that is located on the back of the whale. So this is one trick to separate clicks from untagged whales from clicks of tagged whales that come here all the way to the bottom. And then we have buzzes. So what are buzzes? These are clicks. And this is a buzz. So clicks are discrete broadband sounds, very short, that are used like in bats to echolocate, to locate prey and even differentiate the characteristics of the prey. So they are used for long-range search, while buzzes are used to, when you are very close to the prey, you need many updates because you want to know exactly what the prey is to be able to react very quickly and get that prey in your mouth. So some friends and us tend to compare clicks with acoustic photographs and buzzes with acoustic video. So you get a click, you get an acoustic scene, you make another click, you get the acoustic scene, and then you get the buzz when you get many acoustic scenes very quickly. So what happens? That the spacing between clicks, the inter-click interval, gives you an idea of how long the whale wants to wait until receiving new updates, which is related to the distance that the sound can travel in that time. So the inter-click interval gives you an indication of the distance at which the animals are waiting or expecting to or looking for prey. So let's go layer by layer of the information that is provided by these acoustic tags. The first thing that you have is the depth. So the bathymetry sensor, the depth sensor, the pressure sensor of the whale. This is a 3D representation of a dive, a deep dive of a blainbill's big whale. And I would like you to think on this. The animal starts the dive here, is colored by depth. So in dark blue is when the animal is at the surface, it goes down, 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 down. It starts moving around, looking for prey, and then it ascends. But blainbill's big whales, in general, studied big whales, they don't ascend in vertical as pilot whales do or sperm whales do. They do a low pitch ascent. So I would like you to think on this while I keep talking and while you still listen. Why an animal that is diving deep and that, you expect that animal to want to go up very quickly, why will this animal make a low pitch, a very low inclination ascent? While you think on that, we go to example dive profiles of pilot whales. So these are 3 example whales, and in this case, the color of the dives is the speed of the whale, the vertical speed in meters per second. So you have that whales here, very deep, they are doing sprints. They are going really fast. The fastest speed was 9 meters per second, which is 18 knots, which is something like 36 kilometers per hour. Sorry, I don't talk in miles per hour. And but 18 knots is something like 18 miles per hour, nautical miles per hour. So what you see is that they do series of dives with a mean of 5 dives per series. Then they have periods at the surface, and the maximum depth and duration of the deep dives was 21 minutes and something like 1,000 meters. Here, this vertical line is marking the sunset, when the sun, the day and the night. So you see that in the day, they are going deep and they are doing very few shallow dives. In the night, they are doing both deep dives and shallow dives. And then we put sound on, and this, is the behavior of blainbills. So in contrast to pilot whales, they don't do series of deep dives. they do isolated deep dives. The depth is very similar, but instead of being doing shallow surface time between series of deep dives, they do series of shallow dives between the deep dives. These shallow dives can be down to 400 meters, and they can be 20 minutes long. So the maximum duration and depth of Blainville's Victory has been 70 minutes and 1,600 meters. In Cuvier's is 2 hours and 3,000 meters. So these are really extreme divers, extreme deep divers, big whales. So the time that they spend at the surface between dives, on average is, well, the most is 2 minutes. So when they come up after a dive of 1,000 meters, they only spend 2 minutes at the surface. So now sound is going to tell us what the animals are doing in these dives. In the case of pilot whales, when we put sound, here now the circles are the buses, so the prey capture attempts. And again, this line is marking the difference between day and night. This is the time of the day. So you see that during the day, they are going deep, they are doing sprints, and they are trying to attempt very few preys per dive. Actually, 40% of these deep dives have no prey capture attempts. At night, in contrast, they try to capture many more prey per dive, and they forage both at depth and in shallow waters. So let's go deeper into this. Night dives, bimodal foraging. We have here in these multi-panel figures, we have in blue the depth of the whale. So it's going down, in a shallow dive is going something like 200 meters, and the red circles are the prey capture attempts, the buses. Then we have here the ICI, the interclick interval. That is something like 0.2 all over the dive. Then the pitch angle that goes all over the show, don't care about this. The vertical speed, that is never too high. And then the pitch frequency. So you see that the animal goes down. This is a spectrogram of the pitch frequency. And then in the ascent to go up, they have to fluke so that they overcome their floatability. So they have to fluke something like twice every second to go up. So here in green, in this upper panel, we see the depth, the maximum depth at which they are searching for prey, extrapolated from the interclick interval, as we said before. So we see that they were looking from the beginning, they were looking at something like 200 meter depth, and they are hunting at 200 meter depth. During the night, at depth, they also feed on a number of things. But first, so this is the same panel, and we see that first, when they start clicking, they look shallow, so they are like checking if there is anything there, and sometimes they have a buzz here, and then when they pass this shallow layer, they already start looking down deep, and they actually finish hunting down deep. So these animals know where they want to hunt. And why are they going up and down? Because there's this thing, this is a panel, this is the, this from, long ago, since the 60s and before, this graph in the Canary Islands, there's this knowledge of this, that the largest biomass in the planet is not in Amazon. That's the largest biomass on land. But the biomass in the planet is accumulated in something called the deep scattering layer. That is a concentration of organisms that is down there at 600 meters, more or less, in most of the oceans. And this layer is composed of organisms that migrate during the night. They go up to shallow waters where they eat, and then during the day they escape from the visual predators that are near the surface, and they go down to escape from these predators. So during the night they are available for pilot whales to hunt them, but during the night they are not. During the day they are not available, and during the night they are. So that's why pilot whales hunt them at night in shallow waters, but they also want to go and hunt something that is deep. And if they do that, it must be because whatever is down here deep is better prey. Because if you can hunt at 200 meters, why would you spend energy in the locomotion cost of going deep to hunt more or less the same number of prey? If the prey was the same, you will stay up there and spend less energy. So here we think that there are two different types of prey. But then during the day, they do something very different. During the day, they only hunt once or none. And then they are doing these sprints. Remember these sprints in shallow waters, in deep waters. So there's something to consider. That is that the water is a fluid that makes resistance against the movement of an animal. So that drag that the animals have to suffer and they have to overcome, they have to spend energy in overcoming the drag cost of the resistance of the water increases with the speed. It increases to the cube of the speed. So here we have the vertical speed, and we see that during the sprint, the animal is really going up to 9 meters per second. So this is the energy that the whale has to spend during the dive just to overcome the drag cost of the dive. And we see that it increases very slowly until the animal sprints. So in the sprint, they are spending maybe 40% of the energy of the dive. This is the locomotion cost. It increases with the cube of the speed. So you can get the relative cost of the sprint versus the cost of the dive, and it can be very high. So knowing this now, I would like you to bet silently in what are big whales doing. Here we see that pilot whales are hunting deep, and in the shallow dives, they are also hunting shallow. Pilot whales are eating at night in deep and shallow waters, and during the day only in deep waters. Remember that big whales also have deep and shallow dives. So without any further idea, we will hypothesize that they will be feeding both at depth and in shallow waters. But the truth is different. The truth is that they are only eating at depth. So, oh my God, here it is. Remember these dives? So when we put here the acoustic data, we have the buses again as black circles, and we see when the animals start clicking in red asterisk, red stars, and when they end clicking with red stars. So you see that the whales are not clicking until they are something like, maybe at night a little bit shallower, but usually 500 meters down or something like that. They attempt to capture 20 to 30 prey per dive, and they are completely silent the rest of the time. So they are not feeding in shallow dives. So why are they doing these shallow dives? So this is an enigma. And this is both during the day here and during the night. So what we speculated when we saw this is that the whales are doing very deep dives and very long dives. So these long dives are probably exceeding putting their physiology to the limit. So they are using these shallow dives to recover from the deep dives. There's a lot to talk about that, but I have to continue. So are these whales, pilot whales and big whales, sharing a foraging niche? This is the depth, a histogram of the prey capture attempts, and we see that while they feed at depth together, only pilot whales are feeding also in shallow dives. And then are they going to mesopelagic, which is the water depth that is between 200 meters and 1,000 meters depth? That's the mesopelagic zone in the oceans. Or benthopelagic, that means close to the seafloor. Well, we can use the clicks humans use sound as an echo sounder to know where is the seafloor, because you receive echoes from the seafloor, and then you see a band, there's the seafloor. This is how we are used to see these echo sounders. Now let's use the clicks of the whale emitted by the whale and the echoes received on the tag as a bioecho sounder. So you have the clicks of the whale put here in vertical, and these are the echoes of the seafloor received at the tag. So we know where there is the seafloor. Moreover, we also see where are potential preys, because a click has echoes, and the next click has another echo, so a target in the water makes a line of echoes that as the whale advances is going towards the target, and so the echoes are coming closer. And these are these lines that you see here. So you have echoes from potential prey, and now we are doing some more research on to see which ones of these potential prey are actually being eaten. So here we have a dive profile, and we see that when the whale is here, it's going to the deep scattering layer, and look at how many potential preys are there. And the whale is choosing, because there are hundreds of potential targets, and the animal is only eating or trying to capture a few of them. Further, it goes deeper, it goes below the deep scattering layer. So we think that they are trying to hunt the predators of the deep scattering layer that are attacking the deep scattering layer from below. So the whale crashes, goes deeper, and captures those animals. So we put the echoes in as black dots near the dive profiles, and we see that this is a blainbill's big whale, and the whale is going close to the seafloor, while in the case of pilot whales, they are going far from the seafloor. Of course, we have to do more analysis to see if this is true in all pilot whales. But then, what about social foraging? Are they eating together? For that, we put tags in more than one whale. And we see that pilot whales sometimes dive together. Now here, the colors are different whales tagged in the same group. And we see that pilot whales, that are these, can dive together, like here, or completely separate. So they have a very variable synchronization of the dive profile, while big whales have extreme synchronization. They actually synchronize 98% of the dive profile and 99% of their vocal behavior. So when they go down, they go together, and they start clicking together, and they end clicking together, and then they ascend in silence. So it's amazing. And then if the group splits, because the groups are not permanent, if the group splits, then every animal does whatever they want to do. They don't keep the coordination. But when they are together, they are really together. So you would think maybe it's because they are hunting together. But then we did an analysis of the distance between the whales, because if a whale clicks here, the click is received by the tag on the other animal. And the same with the other animal clicks being received on this tag. So you can, by the time that it takes from one click, from being produced by one animal to travel to the other animal, we can see how far they were. So we see, sorry, there are some things in Spanish. This is the dive profile of 3 pilot whales in different colors, A, B, and C whales. And this is how they spread during the foraging time. And they went far, like 200 meters far. While blainbills, big whales, 2 animals diving together, start of clicking, end of clicking, and they split something like 400 meters. So they are not hunting together. So why are they feeding together? And now we come back to the question we made at the beginning. And why are they coming up in a shallow angle after going so deep and for so long? And this is the last of the talk, because this is a hypothesis, but we think it explains their behavior. So these are big whales. Big whales are diving very deep and very long. So they need to stay down deep for a very long time to hunt a number of prey. So these are, these animals evolved to go there because there was a big biomass reservoir. So they want to go there to hunt. As evolution kept its way, it made that the niches in the deep waters spread. So pilot whales are going for something big and calorific, and that's why they sprint after it. And one of the prey is actually giant squid. It's one of the prey. But there are many other things down there that are small. So big whales, instead of being, big energetic animals, they are, big, but they are no energetic. They are low metabolic rate animals. So they can stay down there for very long. Remember that a pilot whale will go down and up in 20 minutes, while a big whale goes down and up in an hour, more or less, on average. So to be down there, they need to go slow. If you go slow, you cannot hunt big prey. So you have to hunt many small prey. So you need to hunt many, so that, because they are low caloric value, because they are small and they don't make you run, if you need many of them, you need to stay there long. So you can see how that, the circle push evolution in one way. But then, if you need to hunt 20 or 30 per dive, and you are going down in a group and spreading to find those, your group cannot be very big, because otherwise you couldn't find so many. You will have to spread a lot. So your group are small. That means that you don't have social defense against predators, and you don't have energy to defend yourself in front of a killer whale. So what is the best? To don't be detected by killer whales. So big whales go down, eat. While they eat, they cannot avoid being detectable by killer whales that can track them from the surface. But once big whales stop clicking, if they were going to go up directly, like pilot whales or sperm whales, they will be easily found by killer whales. But because they go in a low pitch, in a low inclination, and they cover on average 1 kilometer between the time that they stop clicking and the place where they stop clicking and the place where they surface, they are elusive. Killer whales cannot find them because they have something like 3.7 square kilometers to find them, to search for them. That means that the interception rate with killer whales is very low. So big whales have been very successful, and yes, they get hunted by killer whales every so often, but it's rare. So that was successful because they have a low Kelvin interval. They are, slow in every sense, but that makes them vulnerable. When there is a novel stimuli like naval sonar, so big whales are known all over the world because there are mass strandings in coincidence with the use of sonar, very intense sonar that is used to detect submarines by the navies of the world, mainly the US Navy, but also many other navies. So what happens is that they arrive to the beach in mass with internal hemorrhages. So they come out bleeding internally from many organs. This has been related with fat and gas emboli. So our interpretation is that the adaptations that they have to undergo to be able of making these deep dives and long dives with a small body, and the behavioral adaptations to avoid risk of killer whales make them enter in a very strong stress reaction when they hear the sonar. They change the physiological mechanisms that allow them to cope with the challenge of diving so long and so deep, and then they die on the beach. So we need to be courteous with them. Thank you.
Thank you very much, Natacha. We're open for questions now. Anyone has questions? You guys have questions? The behavior?
I cannot hear it.
Everybody's clapping.
Oh, thank you very much.
I have there's a question here from John. It says, on the graph with the killer whale, what is the bar chart on the z-axis?
Oh, sorry. Okay, let me see if I can. Can you see the screen still?
Yeah, we can see the screen.
Okay, because I cannot somehow. So let me see if I can. So if you can see it, if you can see it, it is the histogram was the depth of the clicks. Okay, let me.
So C will be the, okay, go back.
Okay, I can go now. The depth of the depth of the clicks is it was a histogram. Sorry, I cannot go back to the presentation. I don't know why, but there was a histogram on the left, and that was the histogram of joining all the clicks of many tags, putting what was the depth distribution of those clicks. So you see in the histogram, if you could see it, if you can remember it, they don't click shallower than 200 meters, and killer whales usually don't dive deeper than 200 meters. Usually. In the Antarctic now, it has been discovered that they can go down to 1,000 meters to steal fish from the hooks of a fishery. But they go down and up in 10 minutes. And what has been seen is that when they hunt whales, they actually need a long chase to subdue, to hunt the prey. So even if they could go down to 1,000 meters, they could never kill a killer whale at 1,000 meters. They have to wait for them to surface, and that's the trick.
So what would be the strategy of the other big whales to avoid predation?
Well, the studied big whales, we have only tagged Blainville's big whales and Cuvier's big whales, and they do the same. They both only click at depth. And yes, killer whales can hear them from the surface, and we expect that they can track them from the surface acoustically until the big whales stop clicking at an average of 600 meters depth. So at that point, the killer whales don't have any cue of where the big whales are, because if another thing you heard in another talk, that killer whales stop clicking, they don't use echolocations when they are hunting mammals because they don't want to give a cue of their presence. So that's why they hunt silently. So they can use, killer whales cannot use echolocation to find the big whales. They have to try to listen for them, and big whales shut up, so they are not heard. That's the thing. So other big whales, like the largest big whales that are 10 meters long, are the northern bottlenose whales. They behave a little bit more like sperm whales, but these whales are larger and they are in larger groups also. They probably have more social defense against killer whales.
Yeah, so we have a question here.
Hi. So the pilot whales, the short fin pilot whales, are very like similar sizes to both the Blainville's beak whale and the Cuvier's beak whales, but are they not prey for the killer whales, and that's why they ascend so rapidly? Like, how come they're not prey if they're such similar sizes?
So they are similar size, you are right. They are between Blainville's and Cuvier's, right there. But I like to compare them like, the motorbikers, they go in their leather suits and they go in a group to a bar and nobody can sit with them, sorry for the expression. So they are like that. They live they form matrilineal groups where all the animals in a group of pilot whales are related via the mother and the grandmother. So they form large groups like elephants, and they have social defense. So actually, pilot whales have been seen mobbing away groups of killer whales. So they can say, no thanks. Of course, if killer whales will catch a pilot whale alone with a calf, yes, they will be prey.
Okay.
Because they are in a large groups, they have social defense. So groups give you social defense, and they give you also dilution of the predation risk. Because if you are many and you have a predator that can only eat one, if you are only one, it will eat you. But if you are many, it will eat one of you. So you benefit by the number of animals.
Okay, thank you.
Thank you. Well, thank you so much. I think you have all our time. I want to thank you for your time. I know you have a very long flight yesterday, and I appreciate you making the time to be here with us. And thank you, everybody, for joining us today. This presentation is going to be posted on our network website so that everybody gets a chance to hear your great work. So, bye, everybody.